Hello, everybody, and welcome to our class on application programming interfaces, or APIs. And our goal is to learn about APIs and apply it to getting RNA-seq data from the ENCODE portal. And so we're going to do that by figuring out how the ENCODE portal speaks. And we use APIs all the time. When we log into a web page or press a button on a web page, we're communicating with the other computer or website. And so it's how that interface talks, and we'll go through that using ENCODE portal as an example, which has just thousands and thousands of data sets. So you can apply what we're going to do today to downloading any of your favorite data, also keeping track of a bunch of parameters about that data that we'll use in the next class to make a sample sheet. So we want that data, but we also want to make sure it's good and reproducible. So we have to take an extra step and learn about application programming interfaces, or APIs. And so we're here in class 2023, classes 05 RNalyses, and we are now moving into RNAseq. And we are starting in 00, which means we're prepping to get the data, is why it's labeled as a 0. And in there, we are doing 01 ENCODE API here. And I'm going to run that code in RStudio. And if you're on the web here at long-non-coding-RNA.io teaching, then we have now moved into RNA-seq analyses here and starting at number 8. All right, so we have to install a new package, HTTR, and just make sure you have some of these other ones. I've already loaded them into my environment. And so again, the goal is listed in here about we want to understand what is an API and how can we use it to get the data that we want. Okay, so first thing is let's go to the ENCODE portal here. And just typing in ENCODE, we'll get right to it. And we'll go to this functional genomics where we looked for our ChIP-seq data. And now here we're going to select for RNA-seq data. We are working with our ChIP data in HepG2. So we want to pick humans and then the HepG2 cell line here. I'm going to go through this kind of quick, so feel free to pause it and go back and look at some of the other filters you can do. And library prep, the type of library we want to be paired end for our RNA-seq data. Okay, so now if we do that, we see that there's 5 samples that match the selection criteria. And oh, look, we can download them. Well, not so fast. So what we can do is also see a report here, which gives us information, because each of those 5 things are an experimental accession that starts with ENCS here. And in there are multiple ENCODE files. So we have one experimental accession that has many ENCODE files inside of it, like replicates and other things. So we can go back to this experimental matrix, which is simpler to look at, where we have 5 samples. If we click on what those 5 are, we can see it's insoluble cytoplasmic membrane, cytosol nuclear, and total RNA. And we're going to grab all of those. But just to point out here, we see the ENCSR label up here. And if we go in here, we can see that there's actually more than one file. So there's 2 read types, and there's 2 replicates in here. So one experimental accession equals multiple ENCODE files inside of it. Okay, so let's go back to our total list of these are the things we want. And so we can click this download button here. And what we get is don't worry, it's not going to just download them. What we get is a URL of how to download them. So we could curl that and use this argument here and download those files using that URL. But we can also in here do select files. So it's just going to get the files matching our selected filters here. And if we download that, this file looks like this is the one we're going to download or start to learn how ENCODE is transferring this information of us selecting experiments into a URL that we can get our own information from. Okay, so then these will be where to download, so we can just wget those and download all the FASTQ files labeled here. And you'll notice the ENCFF annotation here, and that's an ENCODE file for an ENCODE experimental accession, which is the 5 things we've selected here. Okay, so that's just a little background on more on the ENCODE portal. And so if we take a look at this link and just copy and paste this URL, which is actually a good idea to keep track of where your data came from, and we go to this code here, this is what that URL looks like. Sorry, this is for the report. So if we look at the report that has all the information, and this download TSV button, we can actually right click and copy link address. And if you do that, that's the URL you get is this one here. And so this is saying, tell me for those 5 experimental accessions, tell me all the ENCODE files associated with them. And things that we can start to see in here are like experimental control type, total RNA-seq replicates, title is equal to that. So we can see that there's actually code inside of that URL. And the reason we're doing this is if we actually look for the MD5 sums, which will say if we downloaded the files properly from ENCODE, they don't have that button here. And so in columns, you can click things and get things like controls here. We don't need that for our RNA-seq, but for ChIP-seq, you might want that for the inputs. All kinds of different things you can sort by, but MD5 sum is not one of them. So this could be a problem for you, and we will find the solution, which is how does ENCODE think to go find this? And when I click a button, what am I adding to that URL? And then we're going to make our own, and we're going to sort of use our own custom speaking to the ENCODE API so that we can get our MD5 sums that are so important to make sure your data is downloaded correctly. Okay, so now we're going to dissect this a little more and then write a short little function to add in MD5 sum here. And so I've outlined this in text here, and take some time if you want to pause and look over this. But we can see what we're going to do we can see right away in here, there's an accession number. And that there's a field equal to accession number here. And so if we wanted to switch to a different accession, we could just paste in a different one here. That could be cumbersome, especially if we're going to do 480 DNA binding proteins ultimately. So you can look in here and see some of this code here that leads to the experimental accession. And that's something to keep in mind here. And then the base of this URL, we have just this generic thing that's saying we're going to make a report on a given experimental accession. Okay, so now what we are going to do is make 2 functions, and they're described there. And the first is a construct query where we're going to essentially just make a URL with the report information. And we're going to need this information when we make our sample sheet. So we're going to ultimately do RNA-seq and do differential expression. And so we want to keep track of the experimental accession and the file names as we go along so we can be 100% reproducible. So really another goal here is to go from taking a URL that anybody can go to and then downloading that data and making a sample sheet from that we can then do our analysis. So it's a bit of front-end work, but this is something you would do anytime you got a new data set, is get the data, build a sample sheet that keeps track of important information about those samples, and then use that in your analyses as factors to compare in differential expression and other things. So that's where we're going ultimately, but we are going to have to take a few steps to get there. And so we're going to make this function construct query. and set it up so that it's just for your experimental accession of interest. So let's say we are interested in this experimental accession. For this one, we can go and get the ENCODE accession number here and get all the information for it is where our aim is. And so what we're doing is we're adding that base URL, which is on all of the ENCODE URLs, and we're going to file format. So we're just borrowing from here. Format equals FASTQ, data set equals experiment, and then the accession. And so essentially we're just making that first part here in text. And the input parameter is the experimental accession you're interested in for this function. So then the fields we want to get are MD5 sum. This is our main goal. We're doing a lot of this work just to make sure our data is reproducible. But this will be handy in the future, especially if you use ENCODE data. But okay, so we want the accession number, we want the read count, the MD5 sum, what it was controlled with, its paired end, paired with, replicate, and target. And we got all that information from this URL we got from the ENCODE website. So we're just making the same format for the URL where we can modify in get MD5 sums for one. So we did have to experiment with this to get that. Was it MD5? Was it MD5 sum? And we could run this and then see if we retrieved information for that column. And we found out that the MD5 sums are stored on ENCODE's website, and they're called MD5 sum, even though there's not a button for them on the website. Okay, so then we're just going to keep adding information to this URL. We're just continuing to make it. And we'll notice that there's this %2f separator here, also in this one here. And so we're just taking this and then putting in our parameter now. This is the main part of the function where we're putting in our accession number that we want, and then the information we want to retrieve for that experimental accession. And then we can imagine doing a for loop and doing this for a bunch of samples at once, just by getting a list of experimental accessions right from the ENCODE website. Okay, and so we are going to talk about map a lot in the next class, but essentially the problem here is there's more than one experiment that we want this information for. Like there was 2 paired ends and 2 replicates made 4 files in a given experimental accession. So we need to map those fields we made here, fields, to a single column. So we're going to basically take a bunch of files, information, and paste all 4 values in one slot. And we're going to look at that more because we're going to have to do that as we move forward in our analyses of compressing the information for things into one row, so to speak. It's really cool, and we'll go over that in detail in the next class. But for now, we're basically going to take those fields, and we're going to have those fields for multiple experiments, and we're going to put them into one unit of memory, so to speak. Okay. And so now we can, that's another key thing here. And the spacers in the URL are an and sign. And now we're just retrieving our URL, which is to paste the base URL with our query, which is what we just made here. So we're essentially just really pasting in a URL, but it's kind of fun to know that you can make your own custom way of speaking to a website that you normally just use buttons on. Okay, so let's run that. And now in our environment, we should have a function. We do here construct query. Okay, cool. So now we need to give it an accession number. And so let's do this test example here. And I put in ENC, this experimental accession. We can actually go, this is handy to do, is go to ENCODE project and then go to here and you can search for any given experimental ID, and this is the nuclear RNA from HepG2 that we're trying to download, is what this sample is. So that's how you can quick check. And we go like that. And now what we have is a URL. See if we can get more of it here. Yeah, the URL for our encode. Here's our accession that we pasted in there. And now we have this MD5 sum field where when we go to download this, if we wget this information, we would get a column with MD5 sum for that file. Excellent. Okay, so we're keeping good track of our data to make sure it's not corrupted. And so really this is the same function here that we just went over. All of this is the same, how to get the URL. And then this is HTTR. It's kind of a good thing to know if you're going to be using this, but at some point we have to go to talk to encode. And this is what we're doing here with HTTR code, just some of the basic functions that we're saying get URL, which we made up above in our construct query that we just went over. And then we're saying there's an error message if it's not one of these file types, and some standard checks here. And then we're saying, we're basically just making error messages here. And then the key function here is content in response that we made. We made a response object here, which is going to get the URL, make sure it can connect to encode, and then we'll get that information and return the information as a text file, skipping the first line, which has the URL in it, and just cleaning the names there. So it's not critical to know HTTR very well, but in our case, we're going to need this thing to execute this code here by making a URL, going and getting the information in that URL, and returning it as a text file. So you could copy and paste this for anything you're interested in. Okay, so now if we run that and we establish this as a function here called encode file info here, then we're going to use this to make our sample sheet. So this is really important that we're saying exactly where we went, what information we had, and then when we do analyses, we can use that sort of core reproducible sample information and take out columns we want to do our analyses with and clean them up. And that's what we're going to do in the next class, is make a sample sheet for our RNA-seq analyses from the information we retrieve here. Okay, so now we have a function encode file info, which also runs construct query inside of it first. So now I've just made that function encode file, and I can run it on an accession I'm interested in, and I can go paste this in encode's website to see what it is. And so I now get this object dat, which has my encode files associated with this experiment accession that we put into the function. We have the MD5 sum. That's what we were really after, so we can check to make sure they download appropriately. Paired end identifier and replicate number. Okay, cool. So we got all the information we selected from our construct query, and now we can run this for any accession in encode for any type of data set and retrieve those key pieces of information, which is really all we need to make our sample sheet directly and reproducibly from a URL. Okay, so we are now going to actually download the files. So we just made a construct or encode file info is for making our sample sheet, but the files on the encode portal, when we selected here, when we're on this view showing the results, if we hit download, we get this files thing. That was the default files. I'm going to go download and click here and say selected files here. If we go, and now if we right click on this, we can copy the link, or you can go download selected files, and we look at it here and we get a bunch of paths to download the FASTQ files for the samples we've selected. And this is why we skipped the line, because this is the URL here that gives you the information of where to go to get these files. But we could wget that URL, or we could run the recommended xargs and download the files here, where it gives us this code to do that. And so if we downloaded files.txt, it would look like this, and we can do the same thing now here in our code, where we can take this files.txt that we just downloaded on the web, and we want to make a new directory. So I'm here in RNalyses, RNAseq 00, getting our data set up, and we're going to make a folder called data and then FASTQ. And I'm actually gonna in data make a new folder called FASTQ2, but I'm gonna use this open new terminal here and make sure I'm in the right directory. Okay, cool. So I'm gonna make directory FASTQ2, which you have up here as FASTQ. I'm just doing this so I can do it in parallel. I'm gonna cd into FASTQ2 here. And then I'm going to paste this wget command in there, and I will have, now if I do a list, I can see this files.txt, and if I cat files.txt to look at it, I can see those the same file that we would have downloaded from ENCODE. So you could just go to that URL and copy and paste it, or copy this and paste it in here and get the same data. So, okay, so it told us how to do this. And so what's going to happen is xargs is sort of bash memory or shell memory. And it's going to skip one line and then run curl and run it on this files.txt. There's some other parameters of curl here if you want to look at them in the manual. And so if I now run that here, I'm going to start downloading FASTQ files into my FASTQ2 folder, and I can take a look at that progress. Okay, cool. I've got a metadata TSV and a files.txt that I just used for the wget, and now it's actually downloading these files. And so what we need to do next is need to rename them, and we're going to do all that and keep the replicate and read number information eventually in the next class. In the meantime, there's an exercise to create a function for encode file info. It's a pretty handy thing we just made. And then create a for loop to upload all of your, to make that report file for all of the samples in the RNAseq thing, instead of doing them one by one. And we will do that as well in the next class, where we will now take the information encode file gives us and file info gives us, and make a sample sheet and start doing our RNA-seq differential expression analyses with this free data we just are downloading now. All right, and with that, thank you and be well.